Okay, good morning and welcome to this package demo from Stefano Mangiola. And I think he said if you have questions during the presentation, feel free to raise your hand and we can deal with that during the presentation.
Amazing, thanks a lot. Welcome everybody. So today I will show you sccomp, which is a Bayesian method for doing compositional analysis from single cell data, and how we scale our analysis to the Human Cell Atlas with the goal of modeling the immune system in the human body and the population. So, of course, I assume we all know what a compositional data is from single cell, but just to reiterate, it's pretty easy. Of course, we assume that the clusters are given in this case, and once you define the cluster for many samples, you have here in the middle, you can count the cells for each cluster, yeah? And so you get a total count, and as you can see, these are actually a real data set. Some samples might have a lot of cells, so we might have captured a lot of cells. For other samples, not so much. So each sample can contain different amount of information, if you will. And the information we want to model underneath is cell proportions, yeah? So which is not observed. But nonetheless, if we sum to one, so we pretend that that's known, you can obviously see the proportional distribution of the different cell types, and the goal is to understand between two conditions whether a cell type is more abundant than in another condition. Just to give some background, sccomp is based on a some constrained beta binomial distribution. So the beta binomial distribution is a distribution that works with counts and underlies proportions. And so it's pretty, I would say it's pretty optimal for this kind of data where you have cell counts that underlie a proportional information. However, it's not compositional. So you can, if you have 10 cell types, you can model this with 10 beta binomials, but the beta binomials do not inform each other in this sense. On the other side, there is the Dirichlet multinomial, which is by definition a compositional distribution, also work with counts and underlies proportions, but is not as flexible. So there is just one parameter that defines the spread of all components of the distribution, whether the beta binomial is not compositional, but on the other hand is very flexible. So each distribution can have a different spread. And I will show you later on that neither this is optimal. So you need flexibility, but also you need compositionality. So what we have done is to constrain the means of the beta binomial arrays to sum to one. So that's why it's sum constrained beta binomial. So we can, it basically has a behavior in the middle of these two distributions. One thing we show in the paper is that we generate data from a Dirichlet distribution in this case with 4 components, and for understanding if the sum constrained beta binomial can approximate a Dirichlet distribution on the contrary of a beta binomial, we fit these 3 distributions. So Dirichlet multinomial, of course, is the distribution we generate the data with, sum constrained beta binomial and beta binomial. So the Dirichlet is in black, our distribution is in red, and the independent beta binomials are in blue. As you can see, you expect the components of a compositional data to be inversely correlated. And so you can see that both of course, the black and the red model this property of the data, while the independent beta binomial do not. So there is no correlation across these components. And if we look at the marginal distribution, you can see that the sum constrained beta binomial pretty much approximated the Dirichlet multinomial. So now that this is a good setup, of course, we can capture compositionality here. There is another important property that for single cell compositional data, the means and the variabilities are correlated, are not independent. And that's why you cannot model this with just one parameter from the Dirichlet multinomial. And so that's what you can do with the beta binomial and also with our sum constraint beta binomial. And what there is no regression line here, but what we do is to put in hyper prior that models this association. So you can see that you can imagine a line that goes through this pretty much linear relation. And just as detailed, often single cell compositional data includes this bimodality. There are always some outliers over there, and sccomp can also model this. So we basically, if you wish so, you set parameters, and we actually model this association as a bimodal with two lines instead of one, basically. And again, just to reiterate, if you use a Dirichlet multinomial, you would, in this case, I will explain a bit better, but overestimate the variability for rare cell types and underestimate for abundant cell types. And this, yeah.
Why is there bimodals? Like, what are those outlier bimodal things?
Yeah, so those are rarer cell types. Yeah. Yeah, so there are not because they're not shown in site of, for example. So they might come from the fact that you don't have a lot of information for some cell types. And we tested this association between mean and variability for RNA, site of, and microbiome data. And you can see that, to some extent, you have for all this technology. And each of the line is a data set. So we have 6 data sets for each of these technologies. So the relationship is definitely there. Okay. Well, this was supposed to appear one by one, but I'm not in presenter mode. Well, I'll guide you through. This is the last slide about this relationship. Again, to reiterate that the Dirichlet distribution, it's really quite important for compositional data, but does not apply for single cell. Here we have done, basically, we take real data, and we fit we model this data with different distribution, and then given that model, we generate the data back. So what you would hope is that the model that generates the data with similar properties, for example, mean and over dispersion. So the model is descriptively adequate for your data. And so what we have done here is to get observed data and predicted data and basically do an empirical QQ plot. And you hope the regression line would have slope 1. And that's what we plot in the y-axis. So the different colors are different data sets, and each point is a cell type. Each data set has several cell types we are modeling. And basically, we would hope the slope to be 1, and that's the case for our distribution. But the Dirichlet multinomial, you can see that has a bias, yeah? So again, is overestimating and underestimating, depending if it's rare or abundant cell types. Good. So that's the basic properties of the model. One thing maybe I will not show detail of, but sccomp is also able to detect outlier probabilistically. This is a framework we developed for RNA-seq, and we translate it here, and does an iterative process where you feed the data, try to detect outliers being quite generous, and you refeed the model, and basically you can calculate probabilities of each outlier being an outlier with a 95, for example, credible interval. But I mean, that's it for that point. If you're curious, I can describe a bit more. Let me, for just change here. I'm not sharing my screen. Okay. All right. So just to give an introduction, show you some code. I'll just go through yeah, sure, yeah. I shall go through the README, but
by the way, sorry, can I interrupt?
Yeah.
I'm not a proper competition, but I mean, what would be the importance of these very rare cell types? Can you just not just ignore them and say, oh, I'm just interested in, let's say, the most representative, let's say, cell types?
Yeah, so this strategy is often used for RNA-seq. You have so many genes that maybe you don't really care to each one of them. But for cell type, that's not the case. So you might have 20, 30 cell types, and sometimes rare cell types, NK cells or Tregs are very, very important talking about the immune system, for example, are very, very important for disease. So you really want to model also rare cell types. That's why a count distribution is needed. If you pretend these are observed proportion, then for rare cell types, you would not be able to model the uncertainty of that proportion. So count distribution is very much needed here. Yeah.
The NDA package has it uses conventional tools from, I think it's based on Bloom, but it then has like a kind of a bias correction. They also have compositionality by having the total count, but then they overcorrect and they have like an additional correction step for the bias.
Okay. We compared to a few models, but not that one. There is Speckle is another one based on Lima. Yeah, we show that we outperform many of those models, but we haven't compared this particular. Yeah, but I would say, if you ask to me, model the data as counts is very important. Yeah, beyond compositionality, probably that's the.
I also would expect that.
Yeah, we have a paper. We have a quite thorough benchmark on that. Anyway, just to introduce briefly, there are 5-6 functions. sccomp estimate, there is this function to remove outlier, which is optional. You can pipe all these functions together. There is sccomp test, where you can define contrast and so on. You can ask the model to generate data from the posterior distribution with replicate. You can predict from the model, and you can remove unwanted variation. I will show you that in a little bit. Obviously, all those are good for visualization, for visualizing the underlying effects. It looks like this. You can give a single cell, rats, and count table, and you declare, let's go to count here. You declare the formula of the composition. I haven't explained, but the sccomp does also differential variability analysis. So you can actually give an arbitrary formula for the variability. So what you expect the variability to be affected of. And you give sample and count if you have count data. This bimodal association, you can turn it on, and other cores and so on and so forth. The result looks like this. So you have your cell groups. The parameter in this case is a pretty simple model. You have type, cancer, and the intercept. And you have your effects here and your statistics. And also, if you model the differential variability, you have all those for the variability. So V underscore and C underscore is composition. Proportional for, okay, yeah, this is very detailed, but the effects are in the linear scale. So, the logistic function is not that easily to interpret, is even harder to think about than a log. So we gave a function to actually convert these effects in the unconstrained scale to actually differences in proportion. Of course, differences in proportion are not linear. So if you get the same difference on the close to 0 or close to 0.5, have very different meaning, yeah? But everyone can interpret those as they wish. We have few visualizations. So you can visualize your data, yeah, as a box plot and color by significance. And we do a couple of things that are quite useful. In this case, we identified outliers, so it's not that easy to see here probably, but you have red dots where you have outliers. Let's see if I can point to some. So these are 2 outliers according to the model. So they cannot be easily explained by whatever model you're using. Yeah? So you can choose to remove them if you want to be parsimonious about that. And also this blue box plot is the generated data from the model. So this is quite important because if the blue box plot overlaps with your data, meaning the model is kind of descriptive of your data. Yeah, it's a bit like you cannot fit. You can fit a normal distribution on a gamma, but if you ask the model to reproduce the data, those two distributions will have very different shapes. And so this is a check that the model is interpreting the data well. Sometimes you have, for example, bimodal distribution, and you can see that the model does not do that. So it's very good to say, okay, maybe there is a parameter I'm missing or something, something more complex that the model cannot handle. So this is a quite good check. You can visualize the effects in this way, where you have credible intervals and color by significance here. And again, this is the relationship of the intercepts I showed you before. And maybe the last thing here, well, no, we have some time. In sccomp test, you can test for contrasts, and you can decide the minimal effect you want to test your hypothesis on. There is this tree, if you know a lemma RNA-seq, there is this trait function. So especially in Bayesian statistics, the probability of an effect relies on a minimal effect, which is a good practice if you ask me. So it's not just an effect is bigger than 0, but it's bigger than a certain amount. Yeah. So that's something you can set up as well. You can do ANOVA, and this is in the Bayesian world is done a bit differently, but basically you have two competing models, and you basically test which is the most likely. Yeah. So this is intuitively easy if you wish. So there is the your H0 model that might not include classes, and your hypothesis model that might include classes, and you are asking which is the most likely. And this is done with the Lu package that compares arbitrary Bayesian models. Yeah, so you can see here the two models that are competing, and then you just call Lu compare of the two fitted model. One of the last thing I mentioned, differential variability. As I said, you can use formula variability type. In this case, you don't assume both conditions have the same variability. You can ask two conditions might have the same mean, but one of the conditions might acquire more variability. For example, you can have, as you age, hypertension or hypotension. So maybe the average of the population is the same, but definitely you get less, you lose homeostasis in that, for example, in the blood pressure. Yes, so that would be one of the intuitive examples that we can make. I think that's it. Yeah, these are more plotting, and this is a plot of differential. So this one is the association of the intercept, so mean and variability. And if you test for differential variability, you will not have just intercept, but you will have, in this case, the condition. And again, these are the credible interval and against the 0. So you can see which are deviates from 0 to be significant. And you can see, differences in composition and differences in variability. And you can do just differential variability if you regress out this association. So if you don't know about this association, also the effect will be correlated. Yeah, so will have not much meaning. If something differentially abundant is also differentially variable. So you need to model regress out the baseline association, and then you get those two independent. Yeah, but in the paper we describe this in more detail. Might be not as intuitive concepts, but basically we can do differential variability because we are careful about that association. Good. Any question? Yes. Yep.
I would like.
Oh, you got first.
Yes. I would like to ask about the cell allocation. Is the sccomp specifically defined for certain species, or like how is the annotation works in the S?
No, it's agnostic to annotation. Basically, you have clusters, and you are measuring how their abundance varies in some conditions.
I think the output you have already annotated some certain cell type.
Yes, because your when you give sccomp the column of your clusters, that could have arbitrary names. So the names are in your visualization as well. You know what I mean? I mean, here I don't have the code. Let me see if I should change my screen again. But I don't know. Sorry, maybe I can show just briefly to you here. You know, when you call, you give this cell group column, and cell group could have any value, could have 1, 2, 3, or T cells, B cells, and so on. I will show you then how the count table looks like. I mean, it could be intuitive, but actually is not showed there. Anyway, so for the second part, I will show you how we extended the model to be able to handle random effects, and how we could use this rich model to model how the immune system is distributed in the human body and how it's affected by sex, ethnicity, and age. So for doing this, we have done a work from very early on, harmonizing a lot of the Human Cell Atlas through cell by gene. And we deliver, basically, at the time we collected 30 million cells. We harmonized the annotation, the counts, and a lot of aspects. And we deliver through this curated Atlas query, is on Biconductor. And basically, it's, again, all following this simple, tidy principle. You see uses underneath databases such as Parquet and so on, but for the user, it's very intuitive. You have 30 million cells in front of you. You can filter the table according to whatever you like, age, range, cell type, organs, whatnot, technology, and you can download it as a single cell experiment ready to analyze, rather than spending months to harmonize all this data. And basically, we took this as our database for analysis, and we built a model that was considering, as I said, the difference property of a sample, but also we could respect the hierarchy of the data, which is quite rich in a way. So for the Human Cell Atlas is this big project here, where is a basically collection of single cell data from all tissues of the human body virtually, and across a quite range of population characteristics across more than 6,000 individuals. So you can imagine the higher level of the hierarchy is the human body. Then you have many tissues, and each of those tissues is contributed by many studies, and each study is contributed by many samples. So you have a hierarchy here that you would like a model to be able to to accommodate. This is not only an opportunity to get almost a foundational model of something across the body, but is also needed from a statistical point of view. As for example, this data has not been put together with a specific design in mind. So you have, for example, the age, which is a very important variable here, that has a different range distribution across organs, for example. Yeah, you collect the prostate to old individual generally, and other organs to younger individual. And these, when different groups have a variable range that do not overlap, ignoring those groups in a hierarchical sense could be dangerous for the estimates. And I will give you some example now. So a famous example is the Simpson paradox, where you have, this is quite simple, but I think, can you see it? It's still very small. You have two groups, yeah? And let's say you don't know, or you ignore those groups. And because those groups are shifted in, let's say, the x coordinate, let's say age, in this case, you could have paradoxically a slightly negative relationship. Instead of using random intercept, modeling these two groups independently, and you will see that actually the relationship is strongly positive. Yeah, is this intuitive? And I've seen this happening in practice, not in this study, but in others, so it's actually real. And this is a random intercept, where you assume the effect of age on something, let's say cell proportion, is the same. Yeah? You could have even a richer model, where you could have random effects rather than random intercepts. So for example, this is ignoring the groups, but if you assume that each group can have a different effect, let's say, for example, immune cells as we age would get more abundant in the brain, but less abundant in the heart. You leave these parameters to be estimated by the model. And then let's say these are 3 organs that behave differently, and you might have an overall effect, which is kind of, if you want, the consensus of if any of these 3 relationships, which in this case is slightly negative. Yeah. So those are, the random effect that you can use generally. This is a small detail, but it's just to give some practicality to how we extended the model, at least in a Bayesian context. So normally you have your observed data that is in a linear model. It's quite simple. So it's a product of your design matrix by the effect matrix. And is shaped as such. Your design matrix is sample on the rows, and your effect matrix is cell type, in our case, on the columns. And it's pretty straightforward to add random effects in this case. That is just literally a sum with another matrix product. In this case, the design matrix defines your groupings. So in this case, the grouping could be datasets, could be organs. And again, your effect matrix is shaped in the same way as the previous one, with the detail that for if each of the grouping, you have to have a prior that is a multivariate normal to model the correlation between the effect of the grouping. Yeah, this is the only complication probably. And because you have many cell types, you have to have these multivariate normal priors on each of the cell types. And this would be one level. Let's say we have our fixed effect, our data sets, and then we can add another, as I showed before, which are the organs, and it will be just another sum as this one. So it's quite modular in a way you can extend this type of models. Now I will show you some live, let's see, how you can model random effect with the sccomp in a very, very simple example. And then I will show you what result we got from the Human Cell Atlas. We have 10 minutes, yes, so I'll go pretty quick. Yes, for sure. Enough or even bigger? This must be enough, otherwise there is no room anymore. Let's push this back. Okay, so the first block, I'm just simulating some data. This is a very, very simple model with just random intercept, basically. But it gives us the opportunity to visualize the data as well, simply. So this, I was talking about what a count table look like as input of a sccomp. You have the sample, you have well, you must have your cell group, which is the second column. These 2 sample and cell group are needed, and the counts. Yeah. And then in this case, we have our covariates, which is our grouping is group study and group organ. So in this case, we have a 2 hierarchy, 2 levels. Yeah. And the other columns in this case are the effects of the simulation, but not relevant for a sccomp. All right, so let's plot how does this data look like. Again, here the only effect is actually the intercept, but you can see that we have 4 organs. In reality, we might have more like 20 organs that are modeled in the body. And you can see that each organ has a different overall effect. The blue colored here, organ 3, has a bigger proportion of a certain cell type, and each organ is contributed by 2 studies. Each study has its own proportion because technical effects. So the formula that you would specify in this case would be your fixed formula. So this is omitting any grouping. And then you will have a random intercept for your group study and another group for the organ that sits on top. So in this case, it's a nested random effects. And here, time is really a dummy variable, but I just put it here to show you that you can have both random intercept and random effects here. In this case, you're modeling, these at the global level, how time is affecting cell type proportion, and also you assume that different organs have different, so time has a different effect for each organ. Yeah. And we assume the variability is not dependent on any variable. And that's, what did I do? Okay, didn't load the libraries. Well, where are they? Okay. All right, this is slow. All righty. Okay, anyway, so the model has been fit. Yep, and our estimate looks like I show you pretty much before. In this case, we have 2 cell groups, A and B, and we have the effects for each of those cell groups, including the fixed effect and all the random effects. In this case, intercept mostly, but also time for the organ. And you have, again, the effect, and you can calculate statistic with sccomp test. In this case, again, time is a dummy variable, so in this case, it's not relevant. But just to show you the gist, just to define a function for plotting, because we are using this over and over. So as I said, for example, if we do sccomp replicate, we are asking the model to produce the data from the posterior distribution. So what the model thinks the data should look like according to what you have specified. And you can see here that, yeah, we reproduce pretty much what we see on the observed data, and these represent the uncertainty. Yeah. So in this case, we have roughly 100 samples, if I remember correctly, and we are asking 10 draws for each of those samples. We might ask many more, yeah. But these, including all the effects, this how the data should look like according to the model, but we could ask how the data should look like eliminating some of those effects. For example, we don't care anymore about the study effects, but we care about the organ. As you can see, the model captured the fact that the organ 3 has a bigger proportion of the cell type and from the other organs. And you can imagine if you have a very, very complex model, you can just slice the posterior, just observing for age overall, just observing for age across the across each organ, so on and so forth.
So suppose that your ethnicity, like
yes, yes.
Yeah.
Only the correct. Yes, any sub formula of your model, you can decide what to preserve. So you can imagine slicing the posterior with random effect, without random effect, and so on. Yeah. And in this case, yeah. The other thing is a kind of similar concept. You can remove the unwanted variation. So rather than asking the model to sample from the posterior, you can, with the kind of a residual, in a way of thinking, but this is all probabilistic, so it works quite well for a small sample size. But in the same way, you can ask the model how the data would look like, the observed data would look like without some of the effect you don't want. And again, you declare a sub-formula here with the effect you want to preserve. Yeah. So this again is including the noise that you cannot model. That's why you're just removing the unwanted variation. And this is the same example as before. So if the study random effect would not be there, this is how the data would look like. And if you do any linear modeling or RNA sequencing analysis, you might be well familiar with removal of unwanted variation. And this is, again, how the data would look like if you eliminate any random effects. In this case, there is no association because this time is a dummy variable, so you just get the, as you can see, there is no effect across organs. Anyway, this is just how you use basically the code. Is a very, very trivial example, but let me show you in the last 5 minutes how this works if you scale this up to thousands of samples from the Human Cell Atlas. What should I do? This is it. And then
the slides of the code available.
The slides, well, I will make them available, yes. Zoom. Here you go. This PowerPoint. Okay, so how the actually formula look like that for our study is quite more complex. So you can see formula composition. We have an interaction between age and sex that we found out, meaning that female and male do not age in the same way across the organs. For example, you might have an immune cell type that increase in the heart of males as we age, but decrease in female. We have a disease factor. Our map is just healthy, so we want to capture the disease effect. Ethnicity, which is a factor of interest for us. Technology, which is a technical aspect. The group is the study, so we are treating the study as a random intercept. And as I said, we have the organ level random effects, which again, we assume that age and sex are dependent on the organs, so are not all the same, and also ethnicity. And the beauty is that we can ask a question to the model, what is the effect on the body and what is the effect on the organ? So in a unique, quite self-consistent model. And also in our case, we model differential variability. We don't assume that as we age, we had the same variability, the same homeostasis in a way, in this case across cell types. We assume yes. Yeah, I mean, for variability, you need a lot of data. It's much harder to estimate difference in variability. So I would not get the variability model that complex.
With this here, you're not assuming that there's any interaction between sex and disease either.
So again?
You assume there's no interaction between sex and disease either.
Not in this model. Yeah. Yeah, I mean, it's a good question. It's it's a good question up to when the model should be complex. Yeah, we decide to stop to the things we were feeling were most associated, but definitely.
And with this, when you're thinking like age and sex, are you doing kind of like a pre and post menopause or menopause?
Not in this case. So yeah, some relationship might not be linear. Yeah? Again, the model could become more and more complex. Yeah, but definitely yes. Some of these might not be linear, and it's good to if you have functions that ask the model to produce the data, because then you can see if your model does not fit the data. Yeah. And non-linearity can be one of those cases, but definitely you could encode that into the model. Yeah, definitely is a good point. So this heat map shows the body effect and the organ effect in a kind of nice way. In this case, this heat map describes the effects. So red is increase and blue is decrease. The effects of age on cell type proportion. So on the vertical, you have organs. On the horizontal, you have immune cell types. And the dots here represent significance, yeah, but we don't focus on that for the moment. You can see here that some of the effects, for example, CD8 naive T cell, as well as CD8 central memory, globally decrease as we age, and this is known fact. But here we can really put a quantitative measure on those. And we have, as you can see, a global effect. So when you look at the significance, you will see a significant level at the fixed effect, as well as some organs that age even more in this sense. And this is also true for granulocyte on the other way. One example that is not like this, so we don't have a global body effect, is T regulatory cells, and we show that the kind of dynamics is very local, organ dependent. as you can see that they increase in brain and muscle, for example, but tend to decrease in lung, well, small intestine, if I remember, yes, and heart, yeah? So that's an example how having a hierarchical model is quite useful. You don't need to split the data. You can build, in a way, a foundation model that try to explain as best as possible the dynamics of the immune system here. For our visualization, especially if you have very complex data, removal, okay, almost done. Let's see, what should I prioritize? Maybe let me, anyway, so this is how you can remove unwanted variation for any effect you like, both random effect and fixed effect. And again, if you assume your data has this, is generated with this model, again, you can, in this case, visualize global body effects of age or effect of age on tissue. Again, just specify a sub-formula of the effect you are interested in. And these are the two examples. So CD8 naive and T. So this is the data with all the variability we don't want removed. And you can see here that very nicely you can see the effect. So a decrease of these two cell types. And on the other hand, the Treg example, we just preserve the tissue effect because we saw heterogeneity. So that's what we want to visualize. And you can see that you can capture heterogeneity here. So the one is at the body level, the other one is the tissue level. This is just to say that is quite nice. Now you can probe the model. You can predict what a immune composition of your body would be for a niche of the population. Let's say in this case is a female of age 40 of European ancestry. Of course, depend on how much data you have to back it up. But the idea is that now you can have a healthy reference on which you can test your hypothesis for your data set. That is more closer to a precision medicine concept. So you can be very specific what you want to test against. And these are the abundances. Yeah, it's as you can see, it's quite complex across organs and immune cell types. These we are removing, just preserving the variability for ethnicity and across 4 dataset, and we show that different ethnicity has quite different immune dynamics in a way. We have done the same for technology. One nice thing is that because all this is probabilistic, so we can not only predict the point estimate, but we can predict the actual posterior. So we can do something like kind of PCAs where we have uncertainty for each. So here we see that some technologies overlap for the immune abundance they tend to detect, and some other technology no. So you have biases across technology for the cell type you can detect, and this would be a technical effect. That's it basically. Quite a lot of people have worked on all these ecosystem and this algorithm that allow this algorithm to work, including Tidiomics community, key people from Bioconductor and from our institute as well, and CSL that is a biotech from Australia that supported some of this work as well. Anyway, just if it was not clear, our lab is growing. If you're interested in any of this work, if you know talented people that want to have the next adventure, please talk to us, talk to me. Thanks a lot.
One more minute, and then we'll start the flash talks at 9:55. Final burning.
Okay.
Thanks a lot.
Ask a question.
Yes.